the unit of life. What is a cell? So when we look around us, there are various living organisms that are basically made up of cells, because having a cellular structure is a defining characteristic of life. Every living organism must contain cell, because what is a cell? Cell is a compartment which determines that the organism is having a basic structure and able to perform is able to perform all the various functions that are needed to live, needed to survive, or needed for life. Because we have seen various unicellular organisms having one cell that have a complete structure and are able to perform all the necessary functions of life. So the first cell was discovered by Robert Hooke. He discovered dead cells viewing some cork cells in a microscope. If I'm not wrong, then it was a simple microscope. After which Anton von Leeuwenhoek discovered the first living cell. Followed by this, Robert Brown discovered the nucleus of the cell for the first time. And after some improvements and advancements in science, the cell theory was proposed. That was by Matthias Schleiden and Theodor Schwann. In 1838, Matthias Schleiden, who was a German botanist, gave out a theory, his contribution in the cell theory, where he examined a large number of plants, and that he observed that all the plants are made up of cells and different kinds of cells, and these cells accumulated to form tissues. Now in 1839, Theodor Schwann made or gave even more important statements and reports. He gave the statement that all the living organisms have cells, and these cells have something known as a outer membrane, which came later to be known as plasma membrane, and the plants have a unique layer presence of cell wall. Now, Schwann also proposed a hypothesis that all the living organisms are made up of cells and products of cells, which was absolutely true. However, this cell theory was yet not able to clarify that from where did these cells arise. So, just to hit the nail, the final touch was given by who? Robert Virchow. He added to the cell theory by saying omnis cellula e cellula, that all the cells in the world, in the earth, arise from pre-existing cells. So there were two important statements in the cell theory. First, all the living organisms are made up of cells and products of cells, and the second, all the cells arise from pre-existing cells, that is omnis cellula e cellula. Here omnis means in everywhere, okay, in the whole world. Omnis, omnipresent, present everywhere. Omnis means everywhere. Cellula e cellula, cell from cell, okay. So everywhere, cell comes from other cells. Or here we have some simple descriptions of cells. Cells have cell membrane and a nucleus which contains the DNA or the genetic material of the organism. There are two basic types of cells, a prokaryotic cell and an eukaryotic cell. And all the cells have some basic organelles, and these organelles are even smaller than cells. Namely, some of the prominent cell organelles in most of the eukaryotic cells are endoplasmic reticulum, lysosomes, mitochondria, microbodies, and vacuoles, most of which we have studied already. However, we are going to look on them in detail. Now, ribosomes are present in both prokaryotic and eukaryotic cells because they are not membrane bound, and they are also found in chloroplasts in plants and in rough endoplasmic reticulum. Animal cells also contain centrosome, which helps in cell division, and plant cells have cell wall. There are different kinds of cells, like RBCs are biconcave, they don't have a nucleus, like WBCs are amoeboid. Amoeboid means they are like amoeba who can change their shape. Columnar epithelial cells, their shape is like column like, and we have longest nerve cell. Moreover, the largest cell in the world is ostrich's egg. It's a single cell and it's the largest. We can see it through naked eye. And tracheate cells, they have tapering ends, and mesophyll cells, they can be round, oval, they can be tubule-like, they can be vesicle or lamellary. Mycoplasmas are the smallest cells, less than their length is only 0.3 micrometers in length, while bacteria are larger than mycoplasmas, 3 to 5 micrometers, and some pleuropneumonia-like organisms, mycoplasmas and bacteria, all form the part of the Monera. Okay. Yes, as I said, these organisms come under Monera and they have prokaryotic cells. They don't have a well-defined nucleus and therefore they come under Monera. Because they are this small, they multiply more rapidly since they don't have such specialized cell organelles. And when we look about bacteria, there are 4 basic types of bacteria. Coccus, Vibrio. Coccus is round, Vibrio is comma shaped, Spirulum is spiral, spiral, and Bacillus is rod like Lactobacillus. And all the prokaryotic cells have a cell wall surrounding the cell membrane, except in mycoplasma. It does not have a cell wall. Cell membrane, it has a cell wall, but it does not have a cell wall. It has a cell membrane. Every cell has cell membrane, and every prokaryotic organism also has a cell wall. However, except mycoplasma. And there's one more specialized structure you can see in prokaryotic cells, that is circular DNA or plasmid. These small circular DNAs confer different properties to these prokaryotic cells, like bacteria. They can give the bacteria resistance to antibiotics or resistance from environmental factors like heat. These circular DNAs are known as plasmids. And no other organelles like in eukaryotic cells are found in prokaryotes except for ribosomes. The infoldings of cell membrane are known as mesosomes. The cell membrane extends to form these mesosomes, which work as mitochondria for prokaryotes. And here this special structure is mesosomes. They form vesicle, tubule, or lamellae. They help in cell replication, cell distribution, forming of the cell wall, and even respiration. Okay, some extensions are also known as chromatophores, which contain pigments. And now there are three layers that envelop the prokaryote cell. First is glycocalyx. Glyco means sugar. Calyx means coat or cover. Okay, so it's basically a sugar cone that can either be fluidy or slime layer, that's known as, or it can be rigid or becoming a capsule. Second, there is a cell wall, and third, there is a cell membrane, out of which cell membrane is the most flexible and it forms the mesosomes. Now, where some stain, where in the cells, in the prokaryotic cells, where the cell envelope sustains some stains, they're known as gram positive. They take up the gram stain, they are known as gram positive. And if we apply stain to the cell, right, or to the bacterial cell, and the stain goes away, then it is known as gram negative bacteria or cell. Alright. Then bacterial cells may be motile or non-motile. If they are motile, then they would have flagella because they help in motility. And these bacteria arise flagellum from their cell membrane too, and it arises from the cell wall. It may look like it's arising from the cell wall, however it's the extension of the cell membrane. It contains the filament, which is the longest part, a hook that's near the cell wall or the cell membrane, and the base, the basal body. Now besides flagella, there are some other structures on the surface that are pili. Well, it is a tube-like structure that helps in DNA transfer. Fimbriae. These fimbriae are pointed fibers on the surface. They help to fix them on tissues if they are bacteria, and not much role in motility. Alright? Yes. The fimbriae helps the bacteria to attach itself in to rocks in the streams and to the host tissues. Then we see ribosomes and inclusion bodies. So ribosomes are site of protein synthesis or protein factories, we know it already, and they're about 15 to 20 nanometers in size, 2 subunits, 50S and 30S units, which form the 70S prokaryotic ribosomes. Several ribosomes may attach to a single messenger RNA, ribose nucleic acid, which is known as polyribosome or polysome. Inclusion bodies. Reserved material in prokaryotics are stored in inclusion bodies, just like in vacuole. However, these inclusion bodies are not membrane bound. Like phosphate granules, phosphate storing inclusion bodies are known as phosphate granules. Cyanophycine means pigment storing inclusion bodies and cyanophycine granules, and then glycogen granules likewise. Gas vacuoles are found in blue, green and purple and green photosynthetic bacteria. So this is a special point. We can find gas vacuoles in some of the bacteria. Blue, green, purple and green photosynthetic bacteria. So blue and green, purple and green photosynthetic bacteria have gas vacuoles here. And we have eukaryotic cells. This is a plant cell, this is an animal cell, it has a cell wall, it is a chloroplast, it also has vacuole, a large vacuole which is not present in animal cells. It has centrosome, see, centriole and centrosomes, almost the same, and lysosomes. The cytoskeletal the cytoskeletal structures of various arthropods, and the cell wall of fungi is made up of chitin. Cell membrane. Cell membrane was studied in the 1950s, especially the red blood cells, because they don't have a nucleus, so the properties of cell membrane can be easily studied. Okay, in other cells like liver cells, there are also nucleus, endoplasmic reticulum, all the cell organelles that are also membrane bound. So explicitly the cell membrane study was not possible. So the study shows that mostly the cell membrane is composed of phospholipids. Lipids and proteins are embedded in the lipids. So they are arranged in a bilayer, 2 layers. The outer sides have hydrophobic tails towards the inner part. So there is a phosphate head, and the tail is a saturated carbon chain. The head is polar, that is having ionic, and that is interacting with water. However, the hydrocarbon chain is not interacting with water because it's hydrophobic, and some of the proteins are embedded in this layer, some of which are fully embedded and some of which are just on the surface, and some are present as a channel for the transport of substances through the proteins that are not interacting with the plasma membrane, with the phospholipid layer. Alright, some cholesterols are also present in the membrane. And like this. This is a fluid mosaic model. It's quasi-fluid, that means it's half fluid. It's because it can flow, it's made up of proteins and lipids. Alright. Lipids have, they are very small, they are macromolecules with respect to size. And then this layer is known as fluid mosaic model. That's because it is fluid, it can flow, that helps to facilitate various exchanges of material. And why mosaic? Because just like a mosaic painting, we have different pieces with different colors arranged for making a special type of pattern that is a bit irregular but in the most calming way. Right? Just like that, with that analogy, we have various protein parts that are embedded in the bilayer, just like a fluid mosaic, just like a mosaic painting. Okay, we have different types of components that are arranged together like a mosaic painting. That's why this is a fluid mosaic model. We have proteins, we have cholesterols, and of course phospholipid layers. There are peripheral proteins. These are the in the periphery, and then we have integral that are in the layer, that in the middle of the phospholipid layer, and also sugar on the peripheral protein. There are two types of transports, active and passive. Examples of passive transport can be osmosis, and active transport is the sodium potassium pump that we're seeing. Okay. Cell wall is seen in plants. The primary cell wall is capable of growth, which gradually diminishes as the plant grows. This primary cell wall is a bit flexible. It's mainly composed of cellulose. Plasmodesmata connects the cytoplasm of neighboring cells with a middle lamella is a layer mainly of calcium pectate. This is very important. The middle lamella is a layer mainly of calcium pectate, which holds or glues the different neighboring cells together. So this middle lamella is composed of calcium pectate, which glues two neighboring cells together, and if two neighboring cells cytoplasm has to be fused for transport of substances, then it's known as plasmodesmata. With this, we complete almost all of the topics except of endoplasmic reticulum, lysosomes, vacuoles, mitochondria, plastids, ribosomes, and cytoskeleton, centrioles, nucleus, and microbodies. We'll do that in the next video. Thank you.